Okay, next up we have Linnea Gyllingberg from Uppsala, and we'll be talking about beyond neurons. Over to you.
Yes. So hi everyone. I feel a bit like an odd bird here because I'm not. So everyone here, are you like neuroscientists or like computational neuroscientists? No, not everyone. One person. Thank you. So it's us two who are not. But I'm still going to talk about things that I hope will be of interest of you. So my name is Linnea. And the talk is titled Beyond Neurons, Modeling Basal Cognition in Slime Molds, but I will talk a bit about other models as well. So about me, 1 month ago, I defended my PhD thesis in applied mathematics, and the title was The Art of Modeling Oscillations and Feedback Across Biological Scales. So I'm an applied mathematician interested in using mathematics to understand different biological systems. And lately, I've been very interested in what's called minimal cognition or basal cognition, so cognition in organisms without neurons. And my thesis, has 4 papers in it. And the first one, which is like the first one in the thesis, but the last one chronologically, which I'm going to go into details in this talk, is about slime molds. And the second one is about modeling fish movement with neuronal models. And I'm going to talk briefly about that because I thought it might be interesting to you. And then I also look at like ecological models. And I have a 4th paper on how we should actually do mathematical modeling in biology called The Lost Art of Mathematical Modeling. And like the themes of my thesis is really like oscillations and feedback mechanisms in biology, and then we have like the art, which is more about how should we model biological systems with mathematics. So just to briefly talk about modeling fish movement. So I have this paper called Using Neuronal Models to Capture Burst and Glide Motion and Leadership in Fish. It's a long title. But so fish movement is usually modeled. Is anyone into modeling fish movement here? No one, nah. But there's like a huge crowd who are really into modeling fish movement. You might not think that because you're all into neuroscience, but people are very interested in modeling fish movement. And usually when you model fish movement, you just assume that they move with constant speed. But when we ask like, well, how do fish actually move? Well, they move with something called burst and glide motion. So they burst for a few seconds, and then they glide due to water resistance. So they decelerate due to water resistance. And one might ask, like, why is this even important? Why should we care about this? Well, it's because this is a way of quantifying social behavior in experiments. And it's also important for studying the evolution of social behavior. But so as I said, like before, people just assume that they move with constant speed, and those models work quite well for some setting, like when you model big fish schools. But when you model like individual fish or pairs of fish, this doesn't work. So we had the idea of using the Fitz-Unaguma model. So does everyone know what the Fitz-Unaguma model is? Kind of, a lot of people do that. It's the opposite when I talk to like fish movement people, no one knows what it is. But it's this model to, and now I get nervous because you know this for real. I'm not a theoretical neuroscientist, but to model the signaling in neurons, right? Yeah, you kind of. And what we can model here is like how the voltage in the neuron, how it's transmitted in the axon is modeled, and then we have a voltage which is V, and then you have W, which is the recovery variable. And if we simulate this, we get this type of oscillating behavior. So with this model as a basis, we wanted to see, can we create a model of fish movement? And our idea was to alter this model. So instead here we have two variables. We have the internal state B and the speed V. And now this is a non-linear term for the speed state. And that is modeled by some arctan function, but we actually get this oscillating behavior with this model. An important parameter here is C. So phi here is an external input parameter, so it kind of tells the fish how much it should burst or glide. So here we see how the external input parameter affects the period length of the solution, meaning that when C is below 0.95, it will just move with constant speed, and then it will hit the bifurcation point and start oscillating, and the periodicity will be lower as C is higher, so the frequency will increase. But what we were interested in is like, can we model, by using a neuronal model, can we model how fish respond to each other when they swim together? So we extended the model to 2 fish, so now we have 6 variables. We have B1, which is the internal state of fish 1, B2, which is the internal state of fish 2, V1, which is the speed of fish 1, V2, which is the speed of fish 2, and then we have the position of fish 1 and position of fish 2. And what we have here is this function which coupled the fish together, which is defined like this. And the reason we have this sigmoidal function. So it takes in the distance to the other fish, and then it kind of boosts, so it acts like an extra boost on C. So here the important parameter is D. And why do we have this model? Well, it's because fish use something called, they are quite wide peripheral vision, meaning that like when we look in front, we kind of see almost just in front and a bit in the sides, whereas fish can see quite wide behind themselves. So this is a way of like modeling this, because they would actually respond a bit to fish behind. And what is interesting when modeling this is that we get like a very rich swimming dynamics. So here we see the distance between the 2 fish here as a function of time, and this is the speed of fish 1 and fish 2. And we get 4 types of swimming dynamics. So we get this chaotic leadership switching, meaning that the fish will first, they move like this, go back and forth, and then one will be the leader, and after a while they will switch and the other one will be the leader. And they do this chaotically. And then here we have this periodic leadership switching. So we will have one leader for maybe 4 or 5 bursts, and then they will switch, or I think it's 7 bursts here, to the other one being the leader. Or we will have this constant leadership, so it's always one leader and the other one is trying to catch up, so they move like this. Or we will have no leadership, we will just have tit-for-tat swimming, so they swim like this. And this is actually what you see in real fish. So with the simple Fitz-Unaguma model, by altering it, we can actually replicate the swimming dynamics of real fish. And if we just look at like a bifurcation diagram of this model, we can see how the coupling parameter D, which is like how much should you react to the other fish, how does that affect your response? We can see that this actually, we get all these types of different swimming dynamics. So this is what we see here is the maximum or minimum of the distance between the fish. And first we have this region where there's no distance between the fish and they just swim with constant swimming speeds. And then we get up to here and we have chaotic leadership switching, and then we have periodic leadership switching, and then we have one leader and one follower depending on the initial conditions, and then we have constant leaderships or tit-for-tat swimming. So with quite a simple model, we get quite rich swimming dynamics. But this wasn't like my main, what I mainly wanted to talk about. I just wanted to show you this, to show like how I want to think about modeling. I want to like be able to take model from different areas. And that's why I think it's so amazing to be at a conference like this, is that you can learn from different areas and use them to model other things in biology that might be of interest. So what I really wanted to talk about is the slime mold. Has anyone heard about the slime mold? Yes. So what's your name? Sandra. Oh, sorry? Sandra. Sandra and Erik and Sandra. That's great. So the slime mold, which is also called a Physarum polycephalum in Latin. It's in English it's also called the multi-headed slime mold, and in Swedish it's called slemsvamp. And it's not a fungus, even though in Swedish it sounds like that it would be a fungus. But it's a cellular or a unicellular organism, meaning that it's just one giant cell. And even though it's just one giant cell, it has multiple nuclei, so tens of thousands of nuclei. And it forms a network of tubes where nutrient is transported. So here it's zoomed in a microscope, and you can see these networks. And here it's even zoomed in more, and you can see actually this tube. And here is the cells or the nuclei stained, so you can see the actual nuclei even more zoomed in. And it can be quite big. It can be like, yeah, this big even. A very big cell. So why am I interested in slime molds? Well, the reason I really like slime molds is that they actually show evidence of learning. And here I define learning as the change in behavior evoked by experience. And this is how it's defined in a paper by Basal et al. And one way they demonstrate learning is through habituation. So habituation is when, like in the morning, you put on your sweatshirt and you kind of feel the sweatshirt, but after a while, you get habituated to that stimuli because you don't, that's like unnecessary information if we would walk around always thinking about how our clothes feel. So we get habituated to that and we don't feel that. And the slime mold can do the same. So here's an experiment where they have the slime mold here and some food here. And then there's a bridge with salt, and slime molds don't like salt. So on the first day, they would try to avoid the salt, but after a while, when you put the slime mold back into the experiment, it gets habituated to the stimuli and it just doesn't care about the salt anymore. And it also shows anticipation. So here's another experiment where they have a slime mold here, food here, and then there's a lamp blinking. And slime molds, they don't like light. And they blink with some frequency. I think it's like 10 minutes or 20 minutes or so on. And after a while, the slime mold actually learns the frequency which the lamp is blinking. So it will go back in time and then go back to get the food and go back in time and go back to get the food, which is quite crazy. It's just one cell. And it also shows abilities of decision making and problem solving. So here, there's food here and there's food here. And after a while, the slime mold, so this is a maze. And after a while, the slime mold will form a network that is optimal in this maze. So the shortest path. And it also can share information. So one slime mold cell and another cell, they can fuse together. So they like fuse together and become one cell. And if this slime mold know the anticipated periodicity of this event or has habituated to the salt or whatever, it will like share it to this slime mold. And then they can separate and then they will both know this information, which is quite crazy, I think. And one interesting thing with the slime mold is, I briefly mentioned it in the slide before, is that it can solve shortest path problems. So in a quite famous experiment, because this paper won the Ig Nobel Prize, or the authors won the Ig Nobel Prize for this paper, they set up, so slime molds love oats. Oats and eggs is their main food in experiments. So they put out oat flakes in the same places as the Tokyo railway system. So they put out oat flakes where the Tokyo's railway stations are. And then after a while, the slime mold formed a network that is as cost efficient and robust as the real rail network, which is quite crazy. So coming back to your talk, Marcus, about like having a parallel computing system. I mean, it's not like it has some sort of brain saying now we should find the shortest path. It just it's self-organized. So like one unicellular brainless organism is as smart as like all the engineers in Tokyo. Quite amazing. On just one task though. I mean, it won't, on this task, it is as good. And why do I talk? I mean, I'm an applied mathematician. Why do I talk about all this? You might ask. I mean, it's cool stuff, but where's the mathematics? Well, oh yeah, first I also had a video so you can see in this experiment. Wait, now I need to find yes, how it actually, so this is very much speeded up. So it kind of like moves around and after a while it settles to like the shortest path between these food sources. So this is how it happens. But this takes like days to actually perform. So this is very much speeded up. Well, because I'm interested in how we can model this. And this was first, models for this was first proposed almost 20 years ago by Tarot et al. And the idea is to model the slime mold as a network. So for example, in this experimental setup, we will have a network of nodes and edges connecting the nodes. And then they model the thickness of the tubes, which is like the thickness in the network between two nodes, I and J. They would change due to the flow between the nodes. So the flow of like particles between the nodes, according to an equation like this. And there are multiple papers for this, where they study this model. It has been proven mathematically that it actually finds the shortest paths, these models, on any network. But what is quite new biologically, or what has been stressed more in the biological parts of slime mold research is oscillations in slime molds. So if we would look at this video, you can see how the slime mold is actually oscillating all the time. Do you see it? That is kind of like the tubes oscillate. And it's also oscillate here. It goes like back and forth like here. Yeah. So you see the oscillations. And in a recent paper by Basal et al. Called Adaptive Behavior Learning in Slime Molds, the role of oscillations, Basal et al. Say that we hypothesize that, as in the brain, modulation of spontaneous oscillations may sustain learning in slime molds. And that I thought was very interesting. So I wanted to look at like, what is the role of oscillations in these types of models, mathematical models, to better understand learning in slime molds. So we proposed a model, and it's called current reinforcement models. That is the name proposed before, and I will explain later why it's called current reinforcement models. And the idea is that we have a network of nodes, and we have three types of nodes. We have sources and sinks and normal nodes. And and then we have a number of particles at each node, I, which is Ni of t. And we have a length between the nodes, which is Lij. And then we have the thickness of the tubes between two nodes, Dij. So it's the thickness of the tubes between I and J. And we have an input, A of t, which is constant here, and an output, minus B of t times N3, so the number of particles here. So I have some inflow there and outflow there. And then the number of particles change due to these equations. So here is a bunch of equations, and it depends on it's a source or a sink. So these are the equations for the number of particles. And then the thickness of the tube at each edge is given by this equation. And that's a lot of equations. But what it basically says is that the more particle flow, the tube gets thicker, and the less particle flow, the tube gets thinner. And then one can ask, like, why is this called a current reinforcement model? Well, it is because so you can have an analogy to electrical networks. So for the slime mold, so we would have the lengthening space in an electrical network would also be a lengthening space in the Physarum, the slime mold. Whereas like Ni is the potential or voltage in an electrical network, whereas here is the amount of nutrients, some sort of food transported around. And then this term, Ni minus Nj divided by the length, is the conductivity for an electrical network, whereas for the Physarum is the thickness of tube. And then Q is some sort of reinforcement intensity, and then this would correspond to the tube expansion rate. And lambda is the conductivity decrease rate of the tube to cable. So that's why what is actually reinforced in this model is this. So it's reinforced based on the current. So that's why it's called current reinforcement model. And that's also the current we have here. So what happens if we simulate this model? So depending on if we have so here is like the network. Now we start by studying it on really simple network. So a regular graph. And when 1, 2, 3 is the shortest path, so this length is shorter than that length, we will see that the input and in the top we have an input and output. So it's A of t, and the other one is minus B of t times n3. They kind of, first it's 1 and the other one is minus 1/2, but then it's self-organized. So the input rate is plus the output rate is 0. So it's 1 and minus 1. And then it quite fast, now I call it the conductivity, but it's the thickness of the tubes. Quite fast, we will have that the conductivity will be 0 on the longer paths, whereas the conductivity will be 10 on the shortest path. And there we see the same, but when the other one is the shortest path. So it eventually it's stabilized, and we have that the shortest path will always be the solution to the system. That's where all the flow will be. And we also show this mathematically in this paper where we study the different equilibrium depending on the length of the edges between the nodes. And we have like when this one is the shortest, this will be the solution. And then when they're equal length, we will have a fully connected graph, and then this will be the solution. But I said I'm interested in oscillations. Why didn't I mention oscillations before? Well, because first we wanted to formulate the model where we actually explicitly define the particles, because that was lacking in the previous models. They never like explicitly defined equations for the particles. So we started with the model with constant input and output, and now we alter it to have oscillating input and output. So now the input is A1 times 1 plus sine of 2 pi phi 1 t plus theta 1 t plus phi 1 minus b and 1 t. And the other one is something similar. And the idea now is that they are not just sources or sinks, they are oscillating between being sources and sinks. So we have two oscillators and the other one are non-oscillating, or we don't have any input and output flow on the other particles. And when we simulate this, we get the same results in the sense that the shortest path will have some conductivity, whereas the non-shortest path will go to, the conductivity will go to 0. So these tubes will disappear. But what happens is that they actually oscillate, and the particle oscillates, and also the input and output oscillate. But still they kind of self-organize. So the input in one node and the output in the other node will always together be 0, even though we don't explicitly define that. But we can see that these curves always cancel out up there in the right-hand corner. And if we look at a bifurcation diagram, so what we are interested in is like, how does like phase difference, for example, between the oscillators, how does this affect the model? So here is a bifurcation diagram where we see the conductivity, and this is for the longer path. So here 1, 2, 3 is the shortest path, which is what we see up there. And what we can see, for example, if we look here, when the phase difference is pi, we get a network like this. So it's the shortest path is much thicker, but it still has some arms to the other nodes. Or if like the phase difference is 0 pi half, where the graph is disconnected. And we can like alter both the phase difference and the amplitude ratio and the frequency ratio, and changing this will get different types of outcomes on the graph. And what we want to do, so this is just like, this was just on a small regular graph. And of course, that is not very realistic. Most line models are very big, even though there are experiments on like shorter or smaller graphs. So we also have simulated this on larger networks. So here we see, here the blue dot and the red dots are oscillating inputs and outputs. And the phase difference is pi between the blue ones and the red ones. And if we look at this, how it looks, and now we will see that the color of the nodes is the number of particles at the nodes. I mean, we can see how it kind of oscillates, and like the tube gets thicker and then it gets thinner, and like the particles move around and oscillates. But this is just like a preliminary result on larger networks. And yeah, so this is a preprint, which is called a minimal model of cognition based on oscillatory reinforcement processes, together with Yu Tian, who sits over there, and David Sumter at the IT department in Uppsala. And yeah, we should update the preprint because we have a new version of it, yeah, there are some grammatical errors in the current preprint. Don't read it too carefully. Read it later. We're going to fix it this week, right? Yes. So next week, read it. Everyone should read it next week. Promise me that. So right now, I want to continue working on this. So I have this, I just recently got a big grant from Gyllingberg Foundation, and I want to continue study cognition through reinforcement and oscillations. And what I think is interesting, there's this, there was this theme issue in Philosophical Transactions B of Royal Society called Reframing Cognition, getting down to the biological basics, where they discussed minimal cognition, so cognition in acelluar organisms in a range of different organisms. And there's also tons of different models to explore. But what I want to do is really to study how can these types of models be used to understand how cognition arises in lower level organisms, so organisms without a brain. And what I'm going to spend the next year, so I'm going to go to UCLA to work with Mason Porter, and then hopefully, and I'm going to continue working with you and with Audrey de Sautour, who provides all the experimental data. So she's this brilliant slime mold researcher at the Animal Cognition Lab at Toulouse, CNRS Toulouse. And that was all I had to say. So thank you.